All right, thank you. Hopefully you guys are well fed and not sleepy. I promise not to put you to sleep. Now I'm going to talk about, obviously what we're doing at Charles River as it relates to the advanced therapeutic space, but more so around how we're adapting the offerings within the portfolio to speed, therapeutic development, and ultimately how we're engaging with the consortia like ARM to provide hopefully data and engagement at multiple levels throughout the space. And I think one of the things that we see specifically in the advanced therapeutic space is that there is always a bit of a tension between needing to move fast using a lot of new technology, but then also trying to bring along the regulatory folks with us and quality folks. So there's this yin and yang, or there's an opposites attract, where you're trying to move fast but deliberately so you can pass regulatory scrutiny. This is a slide that we've been using for the past year or two, talking about how the space in general has evolved over the last several decades. We began life, so to speak, in pharma with a lot of small molecules, which are chemistry, which is transiently modifying biology. We then moved on to biologics or large molecule, where we're producing pieces of biology, which then go and are administered, and the vast majority of times are again transiently modifying biology. And now more recently, in the advanced therapeutic space, many times we're producing entire pieces of biology, which then are administered to patients, and the majority of the time are permanently changing biology, whether this is the elimination of cancer cells or the correction of genes in certain genetic conditions, or even replacement therapies and some of the stem cell therapies. So obviously with that comes a lot of new challenges that we're dealing with. So I had certainly the privilege to be invited to a scientific exchange last November with FDA, MHRA, and EMA as part of the ARM ISCT scientific exchange. And we talked a lot about what we want to see going forward in the area of gene editing as an example. And so I think the way we look at this is we would like to borrow the best parts of some of the programs that have worked well for us in recent past. And obviously one of those was during COVID in the US, we had Operation Warp Speed. And what really came out of that was that we were certainly required to pass regulatory scrutiny for therapeutic development. But what we did there was, in a very good way, was streamline the hurdles that needed to be passed in order to move through those, therapeutic development to clinic and ultimately through, emergency use authorization at that point. But the take-home message was there was kind of a minimum set standard that was set, and as long as you met that, you moved forward. And so we would like to borrow that and take that into this area of advanced therapeutics, especially in places like rare and ultra-rare disease, where we feel like there are limited to no other therapeutic options for a lot of these patient populations, and they can very much benefit from some of these therapies. We still have some work to do, and this is probably my most controversial slide of the talk, but it is something I did present at the scientific exchange. And I think it speaks to some of the work we need to do in collaborative, and for the sake of collaboration within the field. So I'm not picking on these specific products, but I think if you go through BLAs and you look at the CQA tables that are publicly available, a lot of times they look like this. And so I think we need to have an honest conversation amongst ourselves in the field about what kind of knowledge we can share between programs. Because there are some things here that are very, I can understand certain methods, certainly around potency matrices are going to be considered proprietary. And I don't think we would ever ask anybody to release methods by which they're doing that. But I think even things like standards around what you're using for gene editing efficiency, in the case of some of the other ones, and like CAR-T with viral vectors, standard minimums around transduction efficiency, we can probably benefit as a field if we're pulling together some of this data. Certainly, there are entities like FDA and the other regulators, and even to some extent Charles River, that serve as aggregators of a lot of this data, but is very much segmented due to confidentiality amongst the different therapeutic developers. And I think it would benefit us greatly if we could begin to think about ways in a non-competitive environment to figure out what can we share with each other to help advance the field more quickly. When we look at this, and this is data that we presented, and this is from cobbled together from publicly available sources, and we look at gene editing, we're looking at things like identity, potency, cell quantity, and a lot of the safety measures as well. And the two key things that typically come up, as for CQAs and gene-edited products is around potency as it relates to on-target editing, as well as in cell quantity, primarily in viable cell number. We started looking at what are the important aspects of the things that we use to edit these cells, and obviously you're going after identity, you're going after potency, stability, and ultimately specificity. Now, I had a colleague who's here, Sada Kasim, who's at genomic medicine, the genomic medicines part of Danaher. He presented some very good complementary data to this, showing that actually we can get extremely good stability as well as specificity for guide RNA, even if it's at RUO grade. And so while the purity might be well below where we typically like to see it above 80%, the editing efficiency as well as the editing specificity is still 99.99%. And I think this is an important point to make, and it's one that we made to the FDA at the time during the exchange. And Mike Lemecki, who's sitting in the second row, happy to be in the room where we were, a bit badgering the FDA on some of these questions to try to get some answers. Mike wasn't part of that. It was mainly me and Sadek, so Mike was neutral. But we really got into a really good conversation with them about what's more important. Is it purity or is it the specificity of the guides? And ultimately, they made a statement that, yeah, it's probably the specificity because that's what's doing the actual cutting or guiding the cutting of the genes themselves. We ran a quick study internally at CRL within our process development group within our cell therapy CDMO. And we essentially used 4 different electroporators that are all commercially available. One was, it's now commercially available. And then we used a standard Cas nuclease. We edited out TRAC. And what we were trying to look at is whether there was actually a difference between the transfection devices themselves, because as a CDMO, I'm sure you can imagine that we are comparing technologies all the time to try to understand what's the best fit for purpose piece of equipment. And it was really interesting, as you see with the data, but it actually ends up not being so dependent upon which transfection device you're using. It actually ends up being dependent upon, do you get the material in the cells? And this is the quick data from this, and we have larger sets, but just in the interest of time. What you can see is 3 of the 4 transfection devices performed extremely well. The 4th did not in this set of experiments, and I purposefully left this piece of data in to have a bit of an explanation. We actually found out that the buffers that this one was using were not fully optimized, so we actually worked with the vendor to re-optimize the buffers. We ran this study again just before the scientific exchange. I actually got the data the morning that we did it, and we were able to bring TFS4 up with the other 3. So what this really does a good job of showing is it's not really about the transfection device, and it's really about the Cas in this case as well as the guides. You get them in the cell, they do their job. And so this starts to tell us about what's important in CMC when we're going through a gene editing development process for a product. We looked at expansion in these as well. Now, of course, TFS, the transfection device number 4, is lower because we saw a lot of cell death in that one. We were able to recover that once we re-optimized the buffers. But I think in the other part here is generally the 3, the top 3, but even transfection device 4 by day 10 ultimately had a knockout efficiency, which would certainly have passed CQAs for a clinical product. We work internally to try to reduce the bottlenecks by putting a lot of these services, both preclinical as well as clinical and commercial, under a single umbrella. And that serves us to be able to do things in parallel rather than in sequence, because we don't have to align our timelines with another vendor's timelines. The vendor is us. So we've been working with a lot of companies in the CDMO space to look out, we can do everything that you need to do to take you to clinic, but we can do it faster because we own all the pieces and I own all the channels of communication. So we actually did that last year. And we worked with a company that was doing an autologous cell therapy product. They came in with no plasmid, no GMP plasmid, no viral vector, no cell therapy process outside of a T-flask. And this is a very interesting one because we typically get processes in kind of 3 states. We get a process that is, they say, this is my process, this is the only thing I want to do. And we say, okay, and we tech transfer that in and we go. We get a process in that is somewhat developed, but they want to do some additional optimization. And so we say, okay, we're going to do some additional optimization, but we're going to kind of keep your main platform, and we'll tech transfer in most of that. In this case, because they had no process, our common question is what do you want to do? Like, what do you want to dose? What's your dose number going to be? And then once we have that, we work backwards to see what makes sense to use. And so that's what we did here. And in that case, I said, don't even bother transferring us your T-flask process, just let us build it. But the end result here is that without any GMP plasmid or viral vector or cell therapy process to speak of, we signed the SOW mid-February last year. We were able to do all the work required to get them to submit their IND just before Christmas in 24. So this is a 10-month turnaround time to do plasmid, vector, and cell therapy, full process development, training, engineering runs, the whole 9. This is, in my experience, at least 6 months off the typical timeline to do it. We can do it because we can do it in parallel because all the different pieces belong within a single portfolio. And I think, obviously, there's a lot of advantages there. For burn, you have about 6 months less of burn, you get into the clinic 6 months faster, you get moved more into a fast test, fast fail scenario with these types of programs moving forward. We're looking at various other ways we can speed this up, as well as optimize it, specifically on the viral vector side, where we're working with an academic center to look at ways to improve the economics around early phase, viral vector manufacturing, as well as providing several different options around GOIs for the viral vector products as well. I think I've been using this slide on several talks, so I apologize if you've seen it before, but I think the point here is that at least in cell therapy, we have a very good value proposition in a lot of them, where essentially the value prop is take this or you die. And so in that case, you might think for a little bit about whether you're going to do it, but you're going to sign up to take the therapy. And in those cases, you were seeing an instance where demand is far outstripping supply. Last fall, Jason Foster from Ori Biotech and I did a good presentation on this, and we talked a lot about this. In the US, we're addressing about 20 to 30% of the patients that are eligible for commercial CAR T. Globally, that percentage is more like 5%. So we still have a lot of work to do, and the top right graph I think here is very instructive. It's from an IQV report last year, and that is tracking global commercial spend on cell therapy, and that's quite an impressive positive slope there. So we're looking at how we, as a CDMO, need to play in the space as well around technology, which is why we're very active in looking at new technology. But I think one of the things that we focus on quite clearly is how is the technology advancing over time. And so I think what we've seen in the Gen 1, Gen 2 is that we're doing open manual, but in this case, even the G-Rex, as you can see here, can be, in this G-Rex, in this case, is an actual closed G-Rex. But the point is you're moving from open manual, more manual, into closed automated processing, but you still have separate analytics. Generally, when you move to automated processing versus manual, you can get a maximum savings of around 35 to 40%. This is reported in multiple publications, and I'm sure you've seen this in talks many times. Where we start to get some additional efficiencies and cost savings is going to be if we start blending analytics with the process. This might be integrated cell counting. This might be integrated cell counting plus flow cytometry for transduction efficiency. There are different considerations around why you would do this based on cost as well as operational perspectives that we work with a lot of different companies on. We believe the ultimate, Gen 4, whatever Gen it ends up being, is the full melding of process and analytics, and I think we're seeing some companies start to do that really quite well. You know, there's a current and future state that we need to go through here. We're talking about all this stuff now. I think the current state, you know, we're near term. I think we're already moving into gene editing. Obviously, we have products for this, allogeneic potency. The AMT, we've seen the first designation be handed out as part of the Solaris announcement. And then I think we're starting to think about the platform technology designation as well. You know, robotics, there's multiple companies here that are doing it, Solaris, Cellular Origin, certainly Multiply Labs. And then decentralized manufacturing is a big topic, and we've seen some recent movement on that from the UK. I think we'll see some recent movement on, you know, with that with the US. There's a lot of talk about rare, ultra rare versus not that we need to continue as well. But the point is we're having very good conversations. These are conversations we need to have. And it puts us in a good place for the future. With that, thank you for your time. If there's any burning questions, I'm happy to take them. Otherwise, just catch me. I'll hang around. And our room's right outside, so if you need anything else, please feel free to stop by. Appreciate you.